Öncelikle Türkçe bir giriş yapalım. Hoş geldiniz. INFLAMIST platformunun Moleküler Biyoloji Derneği ile ortaklaşa düzenlemiş olduğu İstanbul Nanobodiler çalıştayına hoş geldiniz diyorum ve hepinize başarılı, güzel, faydalı olabilecek bir toplantı diliyorum. Toplantımız 1,5 günlük. Ana konumuz nanobodiler olacak ve de biz de hem gelen misafirlerimizden hem de yaptığımız işlerden bu konuyla ilgili detaylıca tartışmak ve öğrenmek fırsatı bulacağız. So let's continue in English. Well, this is an activity of INFLAMIST platform and also Molecular Biology Association, and that one is dedicated to nanobodies. And it is an activity of INFLAMIST, as I told you, and INFLAMIST is a TÜBİTAK-sponsored 1004 high technology platform. And if I'd like to start with the INFLAMIST, that was one of the 8 platforms which was supported in the beginning as a high technology platform to produce something ready for industry scale production. And this platform was first established in 2018 as a platform of Istanbul University as the leading research university. But it is a collaborative project which is including Koç University, Bezmalem Vakıf University, Bosphorus University, and we also have industry partners, Koçak Pharma and Onko Koçsel. So in total, these academy-industry partnership is aiming to produce novel drugs, to develop targeted treatments and also biomarkers for the treatment and follow-up of inflammasome-mediated inflammatory disorders, mainly familial Mediterranean fever, but also other inflammasomopathies. And this platform, when it was first proposed in 2018, during the preparation phases of the platform, we also organized a workshop. And some of our today's colleagues were attended that workshop as well. And the name of the original workshop was Innovative Biologic Agents for the Treatment of Inflammatory Disorders. So this was the writing steps of this project. And we both aim to develop nanobodies and also pegylated proteins. And we discussed all of the problems, the pitfalls and the solutions about the designing and the development of these novel antibody fragments. That was organized in 2019, and the topics included, again, pegylated proteins, different kinds of nanobodies or heavy chain single domain antibodies from camels, sharks, and their comparison with human antibodies, as well as development of nanobodies plus Fab fragments. So these parts actually constituted our platform, the INFLAMIST platform. And when we organized the INFLAMIST platform, as I told you, we aimed to develop treatments and also biomarkers for familial Mediterranean fever and other inflammasomopathies. But the main aim was development of novel nanobodies plus pegylated drugs plus Fab fragments and also colchicine and biomarkers. As a first example of nanobodies, we selected IL-1 receptor as the target. And for the Fab fragment, the target was colchicine, development of anti-colchicine Fab fragment for those intoxicated persons. And for pegylation, we selected interleukin-1 receptor antagonist, and we aim to develop pegylated IL-1 receptor antagonist. And for colchicine, again, we have been trying to develop some colchicine drugs with longer half-life and also better bioavailability. And also, we are trying to develop biomarkers to predict those patients who are not responding colchicine well and requiring anti-IL-1 treatment. So the first part were dedicated to nanobodies and Fab fragment. Today we will focus on nanobodies, but the original project was including two kinds of antibodies, heavy chain single domain antibodies against IL-1 receptor 1 and also anti-colchicine Fab fragments. And for this project, we have been collaborating with Koç University, Bosphorus University, and Bezmalem University. And also this one is including the Fab fragment, which was organized by Serdar Uysal from Bezmalem University. And our industry partner for these projects is Koçak Pharma. And so far, tomorrow we will discuss with you where we have actually progressed and our drugs or drug candidates are at which step. And the other one was, again, focusing on the colchicine and pegylated drugs. And for pegylated drugs, we selected IL-1 receptor antagonist, which is a natural protein inhibiting the activity of IL-1 beta and IL-1 alpha. But we are using this one as a medication for the treatment of IL-1 driven diseases, which is named as anakinra. But this has a very short half-life, about 3 to 4 hours. And our patients are requiring daily, sometimes twice daily injections, which is really painful. So anything prolonging the duration of this drug would be welcomed by the patient. So the pegylated IL-1 receptor antagonist is one of the project aims. And this one is including Koç University by Seda Kızılel and also İstanbul Üniversitesi. And our industry partner in this project is Onkoilaç, Onkokoçsel. And for colchicine, colchicine is an alkaloid which has been known for more than 3,000 years. So it's been written in Egyptian papyrus and in many other original texts, and it has been used mainly for the treatment of gout and other inflammatory conditions. And one of the areas where colchicine is cultivated is Anatolia, but unfortunately we are still importing colchicine from abroad. So this project is aiming to produce colchicine in Turkey and also to produce higher quality colchicine with longer half-life and better bioavailability using nanotechnologic drug production methods. And this one is a project of our Faculty of Pharmacy, and they are collaborating with Yalova for the production of Colchicum species. We also have some other projects within the platform, mainly aiming to develop methods for preclinical evaluation of efficacy and toxicity of all of the drugs we have been producing. So there are some accredited laboratories, but none of them are dedicated to certain types of diseases. So our aim is to develop methods for IL-1 driven diseases. So that would be a preclinical toxicity and efficacy facility for IL-1 driven conditions. And we are also developing some biomarkers. And unfortunately, colchicine is a wonderful drug, a cheap option for our patients, but when you switch to anti-IL-1 biologic agents, it's becoming enormously expensive treatment. And selection of right patients who are not responding colchicine very well is quite critical, and it's mainly based on patients' descriptions and a few simple laboratory tests showing the inflammatory activity. We are still waiting for some biomarkers to predict those patients who really need a better treatment or biologic treatment. So these biomarkers would be very helpful. So within this platform, we also have two international collaborators. One of them is the Dutch project by Nihaya Mehta and colleagues from Nijmegen. And we are working on the biomarker part of the project with them. And the second EU-supported project is with our colleague Ario, and that one is involving the screening of available libraries for finding anti-IL-1 nanobodies or heavy-chain single-domain antibodies. And we also have another project, this is the last one, which is the project, an independent project, mainly dealing with the social, economic, and cultural issues of drug development and also the problems associated with familial Mediterranean fever. So our team involving investigators, academicians from different disciplines, trying to collect data from patients to understand their unmet need, their problems, and also they are trying to assess the economic burden as well as some legal issues in the novel drug development, especially the problems associated with intellectual property rights. So that's another project. So overall, 11 projects are included in the INFLAMIST platform. So this is an active research network. And of course, one of the main aims of this project is to improve our human resources. So we are trying to increase our manpower who are experienced in molecular biology techniques, experienced in drug development, assessment of these new drugs in terms of efficacy and toxicity, and to provide very experienced, well-trained individuals to the industry as well. So this academia-industry partnership and increasing the manpower is one of the main aims of the project. Therefore, this workshop is one of the activities trying to attract our young investigators to the field and to increase their awareness about these new techniques, the drug development processes, and also to find some new recruits to our projects as well, which are true for investigators at the same time. So I will finish saying a few words about why FMF and why nanobodies. Well, we selected FMF for several reasons. Familial Mediterranean fever is a rare disease. And when we say a rare disease, it means that it is less than 1 in 2,000. It is quite rare throughout the world, but not in Turkey. So it is quite common in Turkey. Although it's a rare disease, the frequency is reaching about 1% in some areas of Turkey. It's usually more than 1 in 1,000. But when you are dealing with a rare disease, there are several advantages in the drug development. So you can run some clinical trials in a smaller patient groups. You have some advantages in the evaluation processes and the development processes. So we are trying to use all of these advantages provided to rare diseases. So that is the main aim. But when you establish the efficacy of any one of these drugs in IL-1 driven diseases, there is a big opportunity. And this opportunity is there are several other IL-1 driven diseases. When it is licensed for FMF, which is actually the case for available drugs like anakinra or canakinumab or rilonacept or even for colchicine, there are several other conditions who are shown to be associated with IL-1. So the inflammasomopathies are including several monogenic conditions, but also several complex, very common disorders as well. There are many different sensors in different inflammasomes, so different sensor proteins may detect different threats or triggers, but at the end, when they are activated, they are producing active caspase-1, which is then activating IL-1 beta or IL-18. So the end product is IL-1, IL-1 beta. So anything inhibiting IL-1 beta is really critical for the treatment of all inflammasomopathies, which are including gout, Crohn's disease, systemic JIA, Still's disease, Behçet disease, obesity, diabetes, cardiovascular disorders, including acute myocardial infarction or any kind of cardiac death, stroke, or neurodegenerative disorders. And quite recently, in June 2023, FDA accepted colchicine as a medication for the treatment of cardiovascular disease. So this is really a very good example of our approach using one of the rare diseases, inflammasomopathies, to develop a drug and then to translate this data or findings into much more common complex conditions involving similar inflammatory mechanisms. And why nanobodies? Well, and first of all, compared to monoclonal antibodies, it is a relatively new technology, and the cost of drug development is relatively lower for nanobodies compared to monoclonal antibodies. And also, maybe more importantly, it is compatible with the infrastructure of our industry partners. That was actually one of the main aims of our platform, because we know that in Turkey there is no drug which is being produced with a Turkish company for monoclonal antibodies using mammalian cell lines. But we have some facilities, some industry partners developing some biologic agents in E. coli platform. So the Koçak Pharma has these facilities, and they are producing insulin using their E. coli facility. So we are trying to use their experience in the production of E. coli platform-based biologic agents. So nanobodies and IL-1 receptor antagonists would be their novel drugs. And the other option is, of course, we have very distinguished international collaborators, which is providing us a very good help. So we have been using 3 different approaches, and tomorrow we will discuss with you our findings. But we used in silico designing. We have been using library screening. We also used immunization with llamas. And we are very happy with the results of the llama immunization, actually. So we love llamas. And this is the last slide. Enjoy the meeting. I hope that would be a very helpful, fruitful, and maybe entertaining meeting as well. Please put your cell phones on silent or vibrate. Please feel free to ask questions. As you have seen, we have lengthy discussion periods. So an hour is a bit unusual for this kind of workshops, which means that we will hear a lot from our colleagues, but there will be enough time for asking questions. Please feel free to ask questions. And if you have any problem, please contact me, Batu Erman, or Gökhan Ardal, or Günnur Ünlü. And we are organizing this meeting in a historical building. Please be respectful in terms of, this building is not the historical one, this is part of it, but all others are actually quite historical. And I think that's all I can say. I'd like to switch to my colleague Batu Erman. He will say something about the other society trying to organize the meeting, and also he will introduce the next speaker.